Hello everyone, welcome to the Science Behind Evidence episode 2. Today we'll be studying about blood identification. Imagine you arrive at a murder scene. There is this reddish brown stain on the floor. Is it really blood? Could it be rust, paint, or tomato sauce? Before DNA analysis, a forensic scientist must answer one simple question: is this blood? Today, we'll learn exactly how forensic experts answer that question. Also, this is the second part of the video. If you haven't watched the part 1, I'll add the link in the description. Do check it out because if you're preparing for NET exam, NT loves to ask questions from the biology of blood as well. Okay, coming back to this video, the techniques for identifying blood stains fall into two broad categories, that is your presumptive test and your confirmatory tests. Presumptive tests include phenolphthalein assay, popularly known as Kastle-Meyer test, calorimetric assay, leukomalachite green assay, benzidine and its derivatives, tetramethylbenzidine or orthotolidine, chemiluminescence and fluorescence assay. Also, why don't we stop after a positive presumptive test? Why do we need a confirmatory test? That is because presumptive tests detect peroxidase-like activity, not blood specifically, because some vegetables, rust, certain chemicals, bleach, they also produce similar reactions. Therefore, confirmation is always required. And that is, in confirmation, you have your Takayama test and Teichmann test. Moving forward, let's study phenolphthalein assay. That is your popularly known as Kastle-Meyer test. Procedure for this test is: place the suspected blood stain on a filter paper. Add 2-3 drops of ethanol. Now, why do we add ethanol first? Ethanol helps expose hemoglobin by lysing the RBC membranes and improving reagent penetration. After that, add 2 drops of working phenolphthalein solution. Wait to ensure no color develops at this particular stage. Now add 2-3 drops of hydrogen peroxide. Now, why hydrogen peroxide? Because hemoglobin behaves like a pseudo peroxide and catalyzes the peroxide decomposition, oxidizing the phenolphthalein into its pink form. That is why an intense pink color confirms the presence of hemoglobin. Next is your leukomalachite green. Malachite green is a triphenyl methane dye. This is what NTA loves to ask. The malachite green is a triphenyl methane dye. Also, the leuco-base form of malachite green is colorless. It is originally colorless. But when you apply the leuco-base to a suspected stain, the heme catalyzes the reaction of leuco-base with hydrogen peroxide, giving a green color. Now, this equation, the leuco-base plus H2O2 plus heme gives green color, is what the reaction components are basically asked by NTA. Also, what I have seen this time, that NTA is asking the procedure, the sequence of these tests. So, you have to learn that also. Also, an interesting fact here is that LMG works on the same principle as the Kastle-Meyer test, only the indicator dye changes. Rest, the chemistry is similar. Now, moving forward, next we have is benzidine and its derivatives, also known as Adler's test. Benzidine and its derivatives oxidize in presence of heme. The oxidation of benzidine is catalyzed by heme to produce blue to dark blue color. The reaction is carried out in acidic conditions. Now the result must be analyzed quickly because this blue color that you have received may turn into brown or fade away after some time. Also very important that benzene and its derivatives are proven carcinogenic and are therefore not suitable for routine forensic testings today. Many laboratories have replaced it with safer reagents like TMB. Next is our TMB and orthotolidine tests. They are basically the same. In tetramethylbenzidine, a drop of TMB solution is placed on a stain followed by a drop of hydrogen peroxide. This gives a immediate blue-green color for positive peroxidase activity, indicative of hemoglobin. Similarly, in orthotolidine test, you place the sample, the cutting or swabbing of the stain on a filter paper, add a drop of TMB solution followed by a drop of 3% hydrogen peroxide. This gives your same blue-green color, that is your positive peroxidase activity. Next is chemiluminescence and fluorescence assay. Chemiluminescence is light emitted as a product of chemical reaction. That is, luminol produces chemiluminescence. Fluorescence assay, on the other hand, requires exposure of an oxidized product, example, fluorescein, to a particular wavelength of light. Fluorescence is then emitted at a longer wavelength. First is luminol. Oxidation of luminol catalyzed by heme produces an intense light, that is your positive reaction, but this can only be observed in the dark environment. Suppose a criminal mobs the floor 5 times. To our eyes, the room looks perfectly clean, perfectly fine. But when investigators switch off the lights and spray luminol, suddenly glowing footprints appear. That's why luminol become famous in forensic investigations. Now, why can luminol detect blood that can be cleaned? Because tiny amounts of iron remain trapped in microscopic cracks and pores. Luminol is sensitive enough to detect these traces. Now, have you wondered, can criminals wash blood away completely? Is that possible? Usually not. Modern reagents often reveal microscopic traces that are invisible to the naked eyes. Next is fluorescein. Oxidized and catalyzed by heme, sprayed stains are exposed to light in the range 425 to 485 nanometer using an alternate light source. Positive reaction shows an intense elevated green fluorescence. Luminol was giving an intense light, the yellowish light. Fluorescein gives a green fluorescence. Now, why use fluorescein instead of luminol? Have you thought it? Because fluorescein works better on vertical stains and does not require complete darkness. Now, moving on to your confirmatory tests, also known as microcrystal assay. Confirmatory tests rely on microcrystal formation and are used to verify the presence of blood in a sample. My main question here is, have you thought why are these called confirmatory tests? Because the crystal formed has a characteristic microscopic appearance unique to hemoglobin derivatives. You are identifying the hemoglobin itself, not just its peroxidase activity. Now, Takayama test's other name is hemochromatin crystal assay. It was documented by Misao Takayama in 1912. The reagent in it is 10% aqueous NaOH 5 ml plus pyridine 5 ml plus glucose saturated solution 5 ml plus distilled water. Procedure for it is: add a drop of reagent on your suspected stain, heat the slide while covering it with a cover slip on a low flame. The result you get is dark pink needle or rhomboid shaped crystals. Very important, this result and your reagent. Even hemochromatin crystal assay, this name is also very important. I know you people get confused in which is hemochromatin, which is hematin. What I do is I just remember the one of them, the other is easily the second one, na? Like that. Now for Teichmann test, it is also known as hematin crystal test, documented by Teichmann in 1853. Reagent here is 0.1 gram KCl, 0.1 gram KBr, potassium bromide, potassium chloride, 0.1 gram potassium iodide plus 1000 ml glacial acetic acid. Procedure for this is: add a drop of reagent on your suspected stain, heat the slide while covering it with a cover slip on a low flame. Now the result here you get is dark brown color rhombic shaped crystals about 10 micro in size. This, even this name also, you have to remember all these things. Very important, we don't use this Takayama and Teichmann test in the laboratories because modern forensic laboratories often rely on immunological methods or DNA profiling, which are more sensitive and provide additional information. Although these tests remain valuable in the teaching or in sub-laboratory settings because they demonstrate the chemistry of blood identification. Also, one bonus question is why menstrual blood is still blood? Because it contains human blood along with the endometrial tissues and secretions. It still gives positive blood test results, though interpretation may require the context. So that's all for today's lesson. If you found this helpful, share it with someone who loves forensic science. Remember, evidence doesn't lie, it only waits for someone who knows how to read it. I'll see you in the next episode of The Science Behind Evidence.